everyone, hope you're all doing good. In this video, we are going to discuss about early embryogenesis. So, in this part, we are going to discuss about the sperm and ova transportation, fertilization and implantation. So, let's discuss about the transportation of sperm first. Okay. So, transportation of sperm. For that, we have to know the testicular anatomy first, right? So, testicular anatomy. So, the testis is the male gonad and it is homologous to the female gonad, that is ovary, and we all know that it is suspended in the scrotum by the spermatic cord. Left testis lies at a lower position than right testis. Okay. The testis parenchyma is protected by tunica albuginea layer. Okay, this is a membrane-like layer, and if you take the glandular part of testis, it consists of approximately 200 to 300 lobules, and in each lobule you will have about 2 to 3 seminiferous tubules. Okay. Now, what happens means these tubules will be anastomosing at the apex to form the reed testis. Okay. Then, efferent tubules will arise from the reed testis and it will be transporting the sperm. Okay, it transports the sperm from the reed testis to the epididymis. And what happens in the epididymis is maturation and storage. Okay. So, you have to know that epididymis has a heavily coiled duct inside it. Okay. And in some men this may measure about 6 meters also. Okay. And there are 3 parts of epididymis as well. You have the head, the body and then the tail. Okay. Now, head is going to transport the sperm from the testis to epididymis. The sperm maturation is going to begin here, and then the body, which contains the coiled duct, which is heavily coiled duct, and here the maturation is going to continue. Okay. And in the tail, it is the transport of the sperm is going to go to the prostatic part of urethra. Okay, prostatic part of urethra. Okay. So, what is going to happen here is there will be final maturation as well as storage will also be taking place here. Clear? So, that is all about the thing that is happening in the epididymis. And after this, what happens? The sperm, usually you have to know one point here, repeating. So, you have to know one point here, sperm usually reaches epididymis after 72 days of initiation of spermatogenesis. Okay. And remember the fact that optimal sperm function requires testosterone levels and this should be normal male range, and they should also have a low scrotal temperature. That is, the scrotal temperature should be lower than the general body temperature. Okay, yes. And know that after ejaculation, the number of sperm in cervical mucus will remain constant for about 24 hours. Okay. Then after that it starts decreasing. However, cervix will be acting as a reservoir for about 72 hours for sperm supply. Clear? Right. And no, there will be about 200 to 300 million sperms deposited by ejaculation, and among this only a few hundred will swim up to the egg. Okay. So, where is the major loss happening? The major loss is happening in the vagina. Why there is a major loss? Because there is expulsion of semen from the introitus. There is digestion of sperm by vaginal enzymes. Third one, there is phagocytosis of sperm along the reproductive tract. Some of the sperms are also engulfed by the endometrial cells, and some are lost into the peritoneal cavity. Okay. So, this is how it goes. Okay. So, there will be expulsion of semen from the introitus, there will be digestion of the sperm by vaginal enzymes, there will be phagocytosis of the sperm along the reproductive tract, and it will be engulfed by the endometrial cells also, and some of them are lost into the peritoneal cavity as well. Clear? Right. Now, let us see this testis anatomy. Yeah. So, this is the testis anatomy where you have the tunica albuginea, the outermost layer, and the numerous septae are going to divide the testis parenchyma into various lobules. Okay. And you have so many seminiferous tubules inside each lobule, and all of them will be coming and joining at the apex, that is reed testis, and from the reed testis you have the epididymis, which has a head, body and then tail, and that will be continuing into the ductus deferens, and it will be going and joining the prostatic part of the urethra. Okay. And after that ejaculation happens. After ejaculation, what happens we have seen. And remember, the liquefaction time for semen is around 20 minutes. Okay, 15 to 20 minutes, right? Yes. Now, after this what happens? There will be a change called capacitation. Okay. So, this is a physiochemical change in the sperm, where the sperm becomes hypermotile and it is able to bind to a secondary oocyte. Okay. So, at which stage the binding is happening? At the stage of secondary oocyte. Then you have to know that capacitation usually takes place in the genital tract only, and this happens for about 2 to 6 hours. There is cyclic AMP dependent phosphorylation with increase in intracellular pH. Okay, so this is what is happening at the cellular level, and remember there is increase in influx of calcium as well as there is efflux of H plus ions. Okay, so this is the physiochemical change that is happening in capacitation. Clear? Right. Now, next one is acrosomal reaction. So, what is acrosomal reaction? See, there are so many layers in the sperm. Okay, acrosome is a layer that is present over the head of the sperm. So, activation of the acrosomal membranes will be there in this acrosomal reaction that will be leading to the release of hyaluronidase, hydrolytic enzymes and acrosin and so many enzymes like that. And what is the role of these enzymes means? It will be digesting the zona pellucida. What is zona pellucida? It is the outermost surrounding layer of the oocyte, right? Yes. Now, what happens when this digestion has been takes place? Zona pellucida has been digested and acrosomal reaction, the acrosome membrane goes and there is release of so many enzymes. So, what happens now is sperm plasma membrane will be fusing with the outer acrosomal membrane. Okay, you have an, see, it will be like this. Okay, you have an outer acrosomal membrane, you have an inner acrosomal membrane, and then you have a sperm plasma membrane. Okay, so think that is a sperm plasma membrane. Now, what happens? There is release of so many enzymes here. So, this layer will be completely gone and these two layers, okay, the layers like, yeah, this layer and this layer will be merging and this will be binding together and it will be coming it as like a opening, okay, so that the contents from here, the nucleus will be paved way to go and fuse with the oocyte nucleus. Clear? Right. Now, let us see about the transportation of egg. So, transportation of egg, this is the tube, right? I am bad in my drawing, just adjust it. I just want you to show the fimbriated end of the tube. Okay, so this is where the ovary lies. Okay, so from the ovary, as ovulation is occurring, okay, say ovulation is occurring, what is going to happen is this fimbriated end of the tube is going to pick up the ovary. Okay, so what is happening? The fimbriated end, it means it just sweeps over the ovary. Okay, it sweeps over the ovary to pick up the egg. Now, the transportation of the egg will be happening in the tube and it will be happening by ciliary motion, tubal fluid, and then muscular movements. Okay. Now, as it is getting transported and say this is the ampulla. Okay. So, this is the interstitium, isthmus, ampulla and fimbriate. Okay. Fimbriate. Repeating. And the fimbriate. Okay. Now, as it reaching the ampulla region, what happens means the cilia that is present here, okay, there will be numerous cilia here. What happens? The cilia will be beating towards the direction of the uterus now. Okay. Yes. So, the cilia in the ampulla beat synchronously, that word is important, okay, beat synchronously towards the uterus. Now, there will also be muscular contractions in the tube, okay, there will be muscular contractions in the tube that will be brought about by the enzymes prostaglandin E2 and prostaglandin F2 alpha, and it will be decreased by certain enzymes like progestins and oxytocin. Okay, so it will be increased by prostaglandin E2 and prostaglandin F2 alpha, and it is decreased by progestins and oxytocin. So what happens after ovulation? The oocyte will be surrounding So after ovulation, what happens means the oocyte and the surrounding cumulus cells, okay, that are present will be staying in the fallopian tube, that is in the ampulla of the fallopian tube, for about 15 to 20 minutes. So that's why what we can tell is the tube also provides a holding action for the ovum, and during this period the changes that are necessary for implantation will be taking place in the endometrium also. So endometrium will undergo the changes necessary for implantation. So implantation, receptive changes we can say. So it becomes receptive for the fertilized embryo. Clear? Yes. So approximately 80 hours in the tube will be spent by the fertilized ovum and the oocyte. Okay, after that, that means 90% of the time will be present in the ampulla alone. So here only the fertilization will be taking place, and the removal of the cumulus cells dispersal will also be taking place in the ampulla. Clear? Right. Now let's discuss about fertilization. Okay. So as we discuss about fertilization, you have to know what is fertilization. Yes, in simple terms, it is nothing but the fusion of male and female pronucleus to form a zygote. Okay. So what is the fertilizable lifespan of oocyte? It is around 12 to 24 hours. Okay. What is the fertilizable lifespan of sperm? It is going to be 48 to 72 hours. Okay. If you see, majority of the pregnancies will be taking place 3 days prior to ovulation, but fertilization can happen anytime before. So this is the ovulation period, means any time before 6 days before ovulation and 3 days after that. Okay. And remember, in the zona pellucida, okay, in the zona pellucida, you have so many ligands are present. Okay. I told you at this time to digest the zona pellucida, in the sperm what reaction will be going? Yes, acrosomal reaction, right? So as the acrosomal reaction is over, it reaches the zona pellucida and it has to destroy it, right? So at that time it binds to so many ligands that are present on the zona pellucida, and especially it is going to be ZP2 ligand. Okay. And after this, the spermatozoa will be entering the perivitellin space. Okay. And it will be entering at an angle. It will not be entering in a horizontal or a vertical manner like that. Okay. In an angulated manner, this is going to enter. And after that, there is a reaction called cortical reaction. So cortical reaction means the head of the sperm contains so many cortical granules. The cortical granules have so many enzymes. So the enzymes of the cortical granules are released. Okay. Yes. So after this happens another reaction called zona reaction. So zona reaction is something where there is hardening of the extracellular layer, and there is also inactivation of the ligands for sperm receptors. Okay, right? Okay, so what was the order it happened? There was acrosomal reaction followed by cortical reaction followed by zona reaction, right? So, this is the order it happened. So, it is A, C, Z. You have to keep it in mind. Clear? Right. Now, after this, say 3 hours after fertilization, the meiosis is completed and there will be release of the second polar body. Okay. Now, what happens after this? The egg will have only haploid chromosomes as well as the sperm will also have haploid chromosomes. Now, both will be fusing together and that will be restoring the diploid chromosomes. That is 23X from the egg definitely, that will be fusing with 23X or Y from the sperm and this will be restoring the diploid chromosomes so that it will become either 46XX or 46XY. Can you understand this? Yes. Now, if the process of fertilization is successfully completed, the term syngamy is used. So, syngamy is a term that is used for successful completion of fertilization. Clear? Right. Now, let me show you this pic, so which will give you some idea on these steps. Okay. Now, let's see this pic where we are able to summarize all the steps. Okay. So, first there is a contact of the sperm head with the ovum. Okay. And then there is a acrosomal reaction where the hydrolytic enzymes are being released. Then the growth of acrosomal process is there. Then fusion of the plasma membrane is there. Then there will be entry of the sperm nucleus. And the sperm nucleus is going to enter at an angulated manner. Okay. So, the other parts are here labeled. Okay. Right. Now, let's speak about cleavage and blastocyst formation. See, cleavage is nothing but a series of mitotic division of zygote. Okay. So, in humans, cleavage is defined as holoblastic. That means the cells will be dividing completely through the cytoplasm, and the next one is asymmetrical. Okay. So, here the daughter cells that are derived are unequal. That means one cell may get more cytoplasm and another cell may get very smaller cytoplasm, and it is also asynchronous. Asynchronous means only one cell will divide at a time. Okay, that is asynchronous. Clear? Can you understand? So, the cleavage that is happening in humans is holoblastic, asymmetrical and asynchronous. Now, after cleavage, what happens means eventually you will get a ball called blastula, and each cell of blastula is termed as blastomeres. Okay. A cluster of 16 to 32 blastomeres together is called as morula. Okay. You have to remember that blastomeres are totipotent up to 8 cell stage. So, what does the term totipotent means? Means each cell can develop. Okay. Each cell can develop into a single complete embryo by itself. Okay. So, that is very important. Totipotent up to 8th cell stage. Okay. Then what happens? There will be fluid that is secreted by the blastomeres and that will be forming a cavity. So, after this fluid formation, this mass will be known as blastocyst since a cavity is formed here. Okay. Now, once after this blastocyst is formed, there will be zona hatching. Okay. Zona hatching means this is a beautiful step where the entire fertilized ovum, okay, that is the stage of blastocyst is going to come out of the zona pellucida. Okay. Then what happens? There will be, if you see the blastocyst, okay, you will have two layers of blastocyst actually, that is inner cell mass, okay, and you also have an outer cell mass like this. Okay. Imagine this is the inner cell mass and the outer cell mass. The inner cell mass is going to become the embryoblast. Okay, that is nothing but the embryo. And remember, this stage is pluripotent. Okay, this stage is pluripotent. Pluripotent means at this stage, these cells can form the different layers of embryo. Okay. So, that is, what are the different layers of embryo? Endoderm, ectoderm as well as mesoderm. Clear? And the outside cell mass will be referred to as the trophoblast. Okay. And this is going to form the fetal portion of placenta. This is going to form the fetal portion of the placenta. And then zona pellucida degeneration will be complete by day 4 of fertilization. At that time, this mass will be ready for implantation as well. Clear? Yes. Now, let's see the series of events that has taken place. So, first there is a fertilized egg, then it will be dividing into a 2-cell stage, a 4-cell stage, then an 8-cell stage, and a completely 8-cell early morulus, then a 16 to 32 cell morulus as such. Then there will be cavitating morula where the cavity is formed and the fluid is getting filled. That stage is known as early blastocyst. And finally, we will get a late blastocyst where you have a inner mass of cells and you have a outer mass of cells. This inner mass of cells is going to known as the embryoblast, which will form the embryo, and the outer mass of cells is known as the trophoblast, which is going to form the fetal portion of the placenta. Is that clear? Right. Now, let's discuss about the implantation process. Okay. So, how implantation is going to take place? So, usually implantation will be taking place on day 6th to 20th of day of the cycle. Okay. So, the crucial histological changes that will be taking place in the endometrium during this time is referred to as decidualization. Okay. What will be happening here means the endometrial fibroblasts will be transformed into specialized secretory decidual cells. Okay, right. So, after decidualization, the endometrium of the uterus is known as the decidua. Okay, so the decidua at which the implantation has taken place. Okay, so the base of this implantation site is known as decidua basalis, and the one which is covering the embryo like a cap is known as decidua capsularis, and the rest of the decidua that is present in the cavity is known as decidua parietalis. Clear? Right. So, what is decidua basalis? Decidua basalis is the site of implantation. Then decidua capsularis is the one that is covers the embryo. And finally, we have the decidua parietalis that will be lining the rest of the endometrial cavity. Okay, rest of the endometrial cavity. Now, two layers, especially the decidua bassalis and decidua parietalis, is divided into stratum, compactum, stratum, bassalis, as well as stratum spongiosum. Okay, so this is how the histologically these two layers are seen. Okay, and remember the endometrial receptive period is between 16 to 22 days of the cycle when she has a normal 28 days cycle. Okay, and this endometrial receptivity is signaled by the hormones like progesterone, prostaglandins, then COX-2 enzymes like that. Okay, and you have to also know that implantation doesn't happen just like an attachment. Okay, it just goes, attaches itself and then it starts growing. It doesn't happen like that. Okay, so there are 3 steps in implantation. First stage is apposition, second stage is adhesion, and the third stage is invasion. Okay, now let's discuss about apposition. Okay, so apposition is something where the blastocyst and the epithelium of the endometrium are having a loose connection or loose interaction. Okay, so it is not very strong, but there is a loose connection that is being established. Okay, so here there will be interaction between the epithelium and the blastocyst. Okay, if you see the epithelium, this will be having pinopods. Okay, so pinopods are nothing but the long finger-like projections that contains the L-selectin ligands. Okay, as well as the blastocyst also contains L-selectin. Okay, now this L-selectin will be interacting with the L-selectin ligands. That is how the loose interaction is established, right? There will also be mucin 1 that is present in the endometrium. So this will be progressively taking the blastocyst to the site of implantation. That is, it doesn't allow the endometrium to get implanted beyond the site of receptivity. Clear? Right. Next one is adhesion. So adhesion, here I have already told you, that is mucin 1. Okay, so this will be acting to cause a successful attachment as well as adhesion, and adhesion through the endometrium will be acting, okay, through the many molecules like there will be many integrins, selectins and cadherins. Okay, and then the final step is invasion. So what happens here? The trophoblastic cells will be penetrating the endometrial epithelium and then it will be invading the stroma of the endometrium. Okay, what happens? There will be increased slices of the stromal cells and that will be burying the blastocyst deeper. Understood? Yes. Okay, and where it will be buried means it will be buried in the stratum compactum layer of decidua. Clear? Right. And finally, there will be ultimate erosion of few maternal arteries also, and as these are eroded, there will be formation of lacunae, and that will be establishing the blood supply. Okay, and after that the further penetration will be stopped by the immunological factor from the mother. Okay, usually the process of implantation is completed by 10th to 11th day after fertilization, or otherwise 24 to 25 days of a last menstrual period. Okay, so the implantation that is happening in human beings in this manner is called interstitial implantation. Interstitial implantation, because the blastocyst is going to bury itself inside the endometrium. Clear? Right. So if you see this pic, I told you there is zona hatching that is taking place. Then there will be so many adhesion molecules present that will be creating a loose interaction which is known as apposition, and with the help of pinopods and so many lichens, there is going to be adhesion which is nothing but the attachment that is going to take place, and finally it will be invading inside the endometrial epithelium and destroying the stromal cells and establishing a contact with the maternal blood vessels. Okay, so this is how the entire process of implantation takes place. Okay, so this is all about early embryogenesis, where we have discussed in detail about the transport of sperm, ova, then fertilization as well as the implantation. I hope this video has been helpful to you. So until we meet in the next video, keep going, happy learning, take care and bye bye.